Okay, so far, everyone get your stuff out. Everyone have their notes? Everyone have a pen? I know. No, the relaxation time was out there. Sorry. We're going to talk about what we did before and what we're going to do today. Okay, so far we spoke about the major components that make up the cytoskeleton, actin, microtubules, intermediate filaments. You guys are all very familiar now with the concepts of nucleation, critical concentration, and how it affects whether a filament is going to grow or shrink, plus and minus ends, treadmilling, and dynamic instability. And so we started talking about the different proteins that regulate the dynamics of the cytoskeleton. And that's pretty much what we did the other day, and we're going to do today and maybe even a little bit tomorrow. Tomorrow? Who's coming tomorrow? Anyone? I'll be here. Not right here. Downstairs. 249. Okay. So we started talking about proteins that nucleate. So that was gamma tubulin ring complex and the centrosome, and all about the growth of the microtubules and how that changes during mitosis. The ARP complex and formins and how they regulate. What? What do they regulate? What kind of cytoskeletal component? Actin, right. And this week's recitation papers are about E. coli and how they basically hijack the ARP complex and some other proteins to form this pedestal that they sit on that helps with adhesion and infection. Then we started talking about proteins that bind to monomer, so actin or tubulin monomers, either to promote growth, so that would be profilin, or to inhibit growth or promote disassembly, catastrophe, thymosin, and staphin. So you should see from how I've organized just this right here, you can start to think about how to organize your information and your thinking for the next exam. Because I'd really rather not spend a whole class organizing it for you. So I gave you the tools last time on how you should organize things in hierarchy, right? And so now maybe start thinking about that with this information, right? And so I'm just going to start you off with this, right? Actin, tubulin, or microtubules, whatever you want. And so then from there, you can start, when you start thinking about all these proteins, you can actually start to think, proteins that nucleate, proteins that bind monomer, same thing. And then start to list them, like that. And so there are categories, and we're going to go through these like in categories. I'm not going to do it all for you. You guys are going to do this yourself. But I'm trying to help you to organize now, not the day or two before the exam. Okay? Right? Because I'm pretty much ignoring intermediate filaments. There was a little bit about it. It's important. They're important. But when we talk about all these proteins that regulate the dynamics, whether or not something's going to grow, whether it's going to be super stable, whether it's going to disassemble, there's just a lot of proteins that you got to keep track of. And by organizing them like this, you'll start to at least have somewhere to put it. And then what we'll do at some point is you'll draw out, and you can even start thinking about this now. Draw out, your actin polymer, maybe some monomers, and you can put things where they go, right? Remember, you've got your minus end and you've got your plus end, and ARP's going to bind over here. You can say ARP, nucleate, and you have your formins, which will bind over here, right? And you can say formins. And then you've got your monomer, and you can say, thymosin or profilin. and then what it does. And so this is how you can start to think about these things. Okay, that's all I'm giving you. So do that as you go along, and then at the end, we can answer questions and help to place things in categories if you haven't figured out where to put them. Okay, so that's what we've done so far. So today we're going to talk about severing proteins, which encourage growth or disassembly, proteins that bind to the side of filaments or polymers, and they will either encourage stability or disassembly, and proteins that bind to ends. I think that's it for today. And there's a theme here, right? It's all about regulating dynamics. Is something going to grow? Is something going to shrink? Is something going to be stable? Is something not going to be stable? And then you need to place it into the broader concept of why the hell does this matter for the cell, right? Who cares if actin's growing or shrinking if you have no idea why, right? So you need to think about, well, what is actin doing? Why would actin need to grow? Oh, a cell's moving. You need to disassemble your actin, send them over to the direction the cell's moving, and then start to grow them. And it needs to create a web of some kind of actin network, and that's probably involving ARP. So this is how I want you guys to think. Okay. Severing proteins. I love that there are severing proteins. I don't know why. I just love the concept of a protein that just chops up everything. Maybe it's because we just came from Halloween. So there's two. So usually when there's a protein with a certain function for microtubule, there's some kind of analogous protein or a protein with analogous function in actin, and that would be catenin. Those who know Japanese can fix my pronunciation. Or gelsolin for actin, right? So this is for microtubules. If you have catenin for 30 seconds, you add it to your microtubules, they start to be chopped up a little. So red is your microtubules. And you can see how they're not one line. What we need here is a zero time point, which should show long lines. And here they're chopped up. Here, 3 minutes, it's down to just little dots. So they can accelerate assembly of new filaments or promote depolymerization of old. So you might be saying, wait a minute, she just said this protein chops things up, something like that. What do you mean can accelerate assembly of new filaments? How would a severing protein accelerate assembly of new filaments? Oh, I like that. It's like cutting a magnet in half. So you end up with two magnets, and you have more ends to add to. So essentially, you're creating more nucleated microtubules by chopping them all up, because it's all whether or not something's going to grow. A lot of it has to do with has that nucleus formed, and is there enough monomer to add to it. And so you've just made a ton of nuclei. Nuclei? I guess. And how is it promoting depolymerization? It's a pretty obvious one. Well, it's chopping them up. So you don't have polymerized microtubules anymore. You just have monomer. If you let the reaction go forever, you'll just end up with tubulin dimers. So this might play an important role in mitosis or meiosis, right? Maybe. What do you guys think? Do you think that there might be a role for catenin in mitosis or meiosis? If you had to speculate what that role might be, just based on your knowledge of what microtubules do. So the way to approach that question is say to yourself, catenin breaks up microtubules, but it also helps them grow. What do microtubules do in mitosis? They just hang out? Nobody wants to raise their hand, huh? Yes? Well, they do. They're involved with chromosome segregation, but go a step further. What happens to your microtubules between interphase and prophase? So interphase is not mitosis, and prophase is the beginning of mitosis. Yes. Formation of mitotic spindle, right? So you're going from one centrosome with microtubules radiating out from the center to duplicated centrosomes with microtubules radiating out from each one. So there's a lot of assembly and disassembly going on, right? Or disassembly and assembly. Okay, things to think about. I just want you always to have some context for thinking about these things. And gelsolin is the protein that severs actin, so similar function, different target. Questions about this? No, it's pretty fun. Okay. So then there are these proteins that bind to the side of filaments, and so they have they can have a stabilizing or destabilizing effect. There are these MAPs, microtubule associated proteins. Those are just exactly what it sounds like. Any protein that associates with microtubules can be called a MAP, but they might have stabilizing or destabilizing effects depending on the protein. So MAP is a family, and within that family are destabilizing or stabilizing proteins. And they can also mediate interaction with other cell components. What kind of cell component might want to interact with microtubules? Huh? Motor proteins, yes. Yes. Motor proteins. What are motor proteins going to do? Transport vesicles, right? And transport other things like organelles, right? Okay. So this picture is showing map localization in axon and dendrites of a neuron. And in this case, the map they're talking about is tau. Well, there's two. There's tau and then MAP2. So you've got green, which is going to be in axon. Is that right? Dendrite? And then orange MAP2. So the question is, do you think MAP2 is going to be stabilizing or destabilizing? Well, in both of these cases, do you think these are stabilizing or destabilizing? These are neurons. I think that you're going to have more stable, these will have more stabilizing functions in neurons, especially in axons, because you need to transport things down the length of an axon. Okay. So, MAP proteins, there's a lot of them. Here, there's tau and MAP2. They bind to the side of the microtubule and project outwards, regulating the organization of microtubule bundles. So, and it's really a size thing. MAP2 looks like this, it binds like this, and it sticks out like this. So it's going to keep the next microtubule at this distance. Tau binds like this, so it sticks out less, and so it's going to organize microtubules more closely. Tau, I don't know who's had any experience with Alzheimer's, but you might be recognizing tau as something that's associated with Alzheimer's. So this right here is, I believe, I think it's from an axon, and it's just showing the microtubules, and see how they're evenly spaced? Each little round circle is one microtubule. It's a cross section. And that even spacing is because of MAP2. And here you can see how it's spaced differently, much more closely together, and here it's tau that's responsible for that difference in spacing. So it's really just, if there's something in your way, you can't get that close, right? Okay, so MAP proteins are regulated by kinases. Oh, we're coming back to this. So whenever I talk about anything, you have to think about regulation. So why would you want to regulate MAP proteins? Well, we'll talk about it in just a minute. They are the next. Oh, good. Okay. Well, so MAP proteins, need to be regulated. You need to control their activity, their localization. And an example is during the cell cycle. You can phosphorylate a MAP protein. And so you know what, I think this slide's in a bad place because I didn't explain that MAP proteins are not only associated with spacing, right? It's also associated with growth or destabilization. So MAP proteins, a whole family of proteins that have many different functions, regulated by kinases. Phosphorylation might inhibit their function, right? So what do you think what do you think the effect of regulation of MAP proteins by cyclin-dependent kinases in the cell cycle might be? Yes? So the most obvious example, right, is it's involved in reorganization of microtubules as you progress into mitosis, right? Cell cycle. So we'll talk about this more as we talk about CDKs in the next few weeks. Here's another thing that MAP proteins are involved with. So this is from a paper that I found that I thought was pretty cool because it had really nice pictures. I think who emailed me is here. Yes. Okay. So MAP proteins, they bind to the side of microtubules, right? And what else goes on microtubules? Someone told me before something that might travel along microtubules. Motor proteins. Okay, so MAP proteins actually compete with motor proteins. And whether or not they're attached to microtubules is going to determine whether or not motor proteins can get by and move things around. So here what they're doing in this paper is they were looking at movement of mitochondria. So mitochondria are going to move around, and it's going to be controlled by motor proteins, and that's going to be regulated by whether or not these MAP proteins, they look at tau, MAP2C, and MAP4, there's a whole bunch of them, whether or not they can be bound to microtubules or not. So in this one, they're looking just at cells where they have stained the mitochondria red. So you can see this even distribution of red and tubulin green. You can see even, I like this picture because here you can see your microtubule, you can see that your cell's in one phase of the cell cycle, and here it's in a different phase of the cell cycle. Do you guys see that? Does anyone see it? Say yes. Right? So here you've got one centrosome, here you've got two. Okay, so that's how it normally looks. And so then they took cells and they made them stably overexpress tau, which is one of these MAP proteins, binds to the sides of microtubules. And what do you see the difference? So they've stained tau green and mitochondria red, right? So they're a reporter in this sense. It's not a reporter assay, but usually you need some kind of readout when you're looking at an effect on something. Here the readout is localization of the mitochondria, right? So here it's kind of evenly distributed in all the cells. What happens here when you overexpress tau? Where are your mitochondria? They're stuck in the center. So, right. And so this is supposed to be plus N-directed motor proteins, so they should be moving outwards to the edge of the cell. And so would you say that that's allowed or inhibited here? Okay, right. So here these tau proteins are binding to the sides of microtubules and inhibiting, see the red? Inhibiting the red from moving outwards. Those are the mitochondria. And so then they also looked at other MAP proteins, MAP2C and MAP4. And this is just them showing that they made stable cell lines. That means that they put the expression vector into the cells and they selected with antibiotics until they had a cell line that was always overexpressing their protein of interest. And you always have to show that when you're doing some kind of experiment because people say, how do I know it's there? So MAP2C is in blue, very nicely expressed, it's blue, and MAP4 And then you can see the same thing here, right? So they're standing for tubulin and mitochondria. And you can see that the red stuck in the middle. Red stuck in the middle. So when they're overexpressing their MAP proteins, it's binding to the site of the microtubules and inhibiting motor proteins from getting by. It's like throwing a bunch of logs on the train tracks. I've been reading a lot of Thomas to my son. He's really into Thomas. Isn't it so funny how cliche children can be? It's like, oh, you're a little boy, you're into Thomas. Big whoop. You know, who knows who Thomas is? Okay, good. Trains. Little boys like trains. And my girls really like princesses. Shocking. Shocking. I'm like, can someone please not be a cliche in my life? I feel like I'm a cliche half the time. Okay, anyways, moving on. Logs on the track. Thomas. MAP proteins. Motor proteins can't get by. Okay. So this whole paper was all about how kinases regulate the activity of these MAP proteins. So what they did is they put in MARK-K2, which is a kinase, right? What do kinases do? Phosphorylate, right? So they made a stable cell line so they've got these cells that are overexpressing tau, and then they put in MARK-K2, and they stain that yellow. It's YFP, so it's yellow. It's amazing the colors that you can use these days. And then they're double staining here with MARK-K2, which is yellow, and mitochondria, which is red. And this is a beautiful picture, right? Because only this one has the kinase in it. And you can see so obviously that overexpression, you still have overexpression of tau everywhere, right? But only here you have a kinase. So what's the role of the kinase in mitochondrial transport here? Right, exactly. MARK-K2 is phosphorylating tau, right? And that's probably resulting in what? Where's tau going to be when it's phosphorylated? Is it going to be bound to microtubules or not? No. Probably not, because those mitochondria are moving along just fine. You see? Can you guys see the difference between this one and this one? Pretty huge difference, right? These mitochondria are not moving outwards. They're stuck right by the centrosome. And these mitochondria are nicely moving outwards. And here they, again, were looking for phosphorylated tau. Because obviously if you're going to say a kinase does something, you need to show that your protein of interest is phosphorylated. And here they're showing when you have overexpression of the kinase, that you've got a lot of phosphorylated tau. They're actually using an antibody against it. So I really wanted to share this with you for two reasons. One, so you could understand that that microtubules are, that proteins move things around on microtubule tracks, kind of like train tracks, and that these side-binding proteins can be inhibitory to that, and that they're regulated by kinases. And so it was a nice example to look at mitochondria movement. Okay, let's move on. Did anyone have any questions about that at all? I always like pictures. Who likes pictures? Who hates pictures? Who has a really hard time with pictures? Actually, another professor was telling me that they had a student who was having a really hard time with pictures, but if you gave them words, fantastic. Isn't that interesting? People are very different in how they learn. Okay, so let's move on. So then there are, so that was all about proteins that bind, the MAP proteins that bind to the side of microtubules, okay? And they can regulate spacing, and they can also regulate assembly and disassembly. So then you have proteins that bind to the side of actin, right? We're gonna cover all the proteins that bind on to both cytoskeletal proteins. So there's a protein called cofilin, and it binds to the side of actin, and it destabilizes. And it does this by forcing actin to twist more tightly, and that weakens the contacts between the actin monomers. So this is just showing the normal actin filament between here and between here. This subunit and this subunit is let's say 74 nanometers. When you add cofilin, it twists tighter, which shortens the distance between those subunits. And when you can just almost imagine if you've got like let's say you have magnets strung together. I like your magnet idea. Magnets strung together, right? And so they bind pretty strongly, and all is well. And if you twist them a little, they can handle it. But if you twist them a lot, you're going to interfere with them interacting, and they'll fall apart. And so you can imagine that's how cofilin is going to work with actin. What's cool about cofilin is that it tends to like to bind to ADP-bound actin compared to ATP-bound actin. So based on that information, you can see that it's going to promote dissociation of older actin filaments, right? Because there's that time lag between ATP hydrolysis, addition and ATP hydrolysis. So ATP-bound actin, those are going to be newer filaments, and ADP-bound actin, those are going to be older filaments. So it likes to bind to ADP-bound actin and promote disassembly of old actin filaments. And that's going to be useful to understand. So to just make a little star next to that in your notes, it's important to understand when we start talking about movement of cells. Okay, so then we've got another protein that likes to bind to the side of actin. This is tropomyosin, and it stabilizes actin filaments. So the other one disassembled, this one stabilizes actin filaments by binding to 7 actin subunits. So here's your actin filament, and here's your tropomyosin, and it's binding to, each one's going to bind to 7 actin subunits. And it can also prevent interaction of actin with other proteins. And the regulation of tropomyosin on actin is important in muscle contraction, and we will talk about that maybe Friday. Okay, questions, comments? Yes?
So cofilin is binding to the side of the actin, like MAP and tau and protein.
Yes, cofilin is binding to the side of actin, similar to all the MAP proteins. Yep. But the important thing to remember, though, is they all have different functions. Some will be stabilizing, some will be destabilizing. So you just need to kind of figure out how you're going to keep that straight. And understanding what they do helps. Like tropomyosin, if it's going to bind 7 actin subunits, that makes sense how it would be stabilizing because it's kind of keeping them together. Okay, practice problems. Okay, we've got 2 here. So we'll go through each one. I'll give you guys a few minutes to talk. Actin filaments in a test tube behave differently than those in a cell. They assemble more readily and have a longer half-life. Why? Provide examples of mechanisms contributing to this observation. So talk to your friends, you have a few minutes, and then we'll discuss. Okay, guys. So it's looking like people are bored by these little talk to your neighbor exercises, because there's so many people just staring ahead. So let's talk about that. How are people feeling about these exercises? Do you find them helpful? Like completely useless? Boring? Awesome? What are you guys thinking? Helpful? Okay. Who thinks that they're a completely useless waste of time and they want me to give them a bunch of things to memorize? I'm just kidding. Who's having, so that for those who are not participating, is it because you're not sure how to address it and you just want the answer, or is it because you're shy and you don't want to talk to your neighbor? One of those two. Who's shy? I'm shy. I don't like to talk to strangers all the time. Kind of. I want you guys to really try hard, even if two people don't know. No one, your neighbor's not going to judge you and think you're dumb. And just waiting for me to tell you the answer, you haven't thought through it, and then you're not going to benefit. Okay, so someone tell me what you think a mechanism behind this first one is, or not even a mechanism. Just give me an explanation for the observed thing. Yep.
There are no disassembly proteins present in the test tube that cause disassembly, which would cause a longer half-life.
So that's one thing, right? So there are none of these proteins. What's in a test tube? What do we say? Actin by itself is in a test tube. There are no disassembly proteins, so they're not going to disassemble more readily. And they're going to have a longer half-life. So that's one part. Does anyone have another thing to add? People who I don't see ever? So that contributed to longer half-life, but assemble more readily. How about that part? Yep. So easier to form a nucleus where? In the test tube or in the cell? Okay, so the question is, in the test tube, what factor is determining the dynamics of actin, whether they're monomer or polymer? Concentration, right? Concentration of monomers. That's the only thing that's going to determine whether you're going to have monomer or polymer in a test tube, because there's nothing else in there. And so what these different proteins are doing, and you guys kind of both touched on it a little, is they are changing that dynamic and changing the critical concentration. And so when you have proteins that are going to nucleate, they'll assemble more readily. When you have proteins that are going to, bind and disassemble, they're going to have a shorter half-life, or bind and stabilize, they're going to have a longer half-life. So there's all these different proteins that are affecting actin dynamics that are in the cell and not in the test tube. Yes?
So you're saying that there must be the protein within the test tube with it that's making it bind more readily?
Well, no. So the question. Let's read through the question, because I think we need to expand on that comment. Actin filaments in the test tube behave differently than those in the cell. Wait a minute, they assemble more readily and have a longer, shoot. Right, okay. The way I was explaining it was as if it was reversed, so my apologies. So yeah, so in the test tube, you would have added those proteins. So sorry guys, I completely threw up. I was explaining this as if in the test tube was nothing and in the cell was all these proteins, and that's why they're different. But they're different in a different way. In the test tube, it's only the concentration that's going to determine whether you have monomer or polymer. And in the cell, there's all these proteins that are going to change the half-life and change their ability to assemble. And actually, for this one, you guys were supposed to tell me what was in the test tube and what it might be doing. So you were kind of right, and you were also kind of right, but I led you astray. So sorry about that. So So what you need to understand is there are no disassembly proteins yet in the test tube. And when you think about assembling more readily, then well, it's just that in the cell there are all these disassembly proteins. Okay, let's do the next one. Cofilin preferentially binds ADP actin and is therefore more likely to disassemble older rather than newer actin filaments. I kind of just told you this, but maybe think about it, look through your notes. Why?
There's a lag between activity and aging. So an older filament will have more ATP. The first question I'll start through reports and those is like I figured it would be simple more So older actin filaments have ADP bound actin because
Okay, guys. Who wants to take a shot at this one?
When the subunits join together and make the polymer, the ATP bound subunits become ADP bound, and so we use the older ones are the only ones with the ADP bound.
Okay, right. So when subunits come together, polymerize to form an actin filament, ATP is hydrolyzed, and so only ADP bound actin. ADP bound actin has been together longer, so it's older, right? Good. Okay, so now we've got another class of proteins. These are capping proteins, proteins that bind to the ends of filaments. And so they tend to be, well, they could be stabilizing or destabilizing, dependent. Yep.
I'm sorry, I don't know if I was the only one who missed that, but it was actually, I don't understand what you said.
Oh, thank you. See how she just did that? Always say, I don't understand what you just said. That's good to say because then I'll repeat it. About cofilin?
Yeah.
Right, because so when you have a monomer that binds together to become polymer, every time you have a monomer bind to the plus end, you have ATP hydrolysis. So your polymer is going to be ADP bound, right? And so cofilin binds ADP actin. There's that lag time between ATP actin binding and hydrolysis. So your older actin filaments are going to be ADP bound. Your newer actin filaments are still going to have some ATP at the plus end.
So you're saying it's because they've been persisting longer in the cell? I mean, the question is because it already says that they're older. I mean, so we know that. I'm just confused.
Cofilin likes to bind to ADP actin. It disassembles the older actin because of that fact. Is that clear for everyone? Okay, good. Okay, so capping proteins. So they just bind usually to the plus or minus end, depending on the protein. This is again a whole category of proteins, and they have a huge effect on filament dynamics, way greater effect on dynamics than side binding proteins, and we'll talk about that for a minute. So what this graph is trying to show you, this is straight out of the book, so you can read the figure legend if it's confusing to you. So what this is trying to show you is that with no capping, your critical concentration is what's pretty much going to determine shrinkage and growth rate, right? Your concentration of monomer. So as your monomer increases, your monomer concentration increases, you're going to have more growth. At lower monomer concentrations, you're going to have shrinkage, what we've talked about in the past. Capping proteins change that. So you're going to actually have at low, let's see. So it basically moves everything higher, stabilizes a bit more in this case. So you're going to have at lower monomer concentrations, it's going to be a little bit less shrinkage because often, in this case at least, it's binding to the plus end. So you can't add any more, so you're not going to have much more growth. See that? So even with high concentrations, you can't add any more because the cap is there. But also, don't forget things come and go from the minus end much more slowly. So you're also going to have so you're going to have a slower rate of shrinkage and a slower rate of growth. So stabilizes. It's stabilizing. So an example is CAP-Z, and CAP-Z binds to the plus end of actin and it stabilizes it. So I just said that one molecule of capping protein has greater effects than one molecule of side binding proteins. Why? Why do you think this might be? So you would say it again? Right. So if your plus end is open and nothing's on it, you can still grow, right? So when you have these different things that bind to the side, then they're going to have an effect, but not as strong as an effect as putting a cap right on the plus end, right? So it's going to have a very strong effect. Like, for example, tropomyosin. That was one that was stabilizing, right? But you need one molecule of tropomyosin to bind seven molecules of actin in order to stabilize that portion. Whereas if you just cap the top, then it's going to stabilize the whole filament. So I worked out the stoichiometry for you guys to kind of illustrate or explain this a little bit better. So the effects of proteins on filaments are dependent upon stoichiometry in a way. So what do I mean by that? You guys thought you were in chemistry for a minute, didn't you? Yeah, you're not. But it's really all chemistry, isn't it? Kind of. How many molecules? This is about how many molecules of regulatory protein are required per molecule of actin tubulin to have the desired effect. So an example is you need one molecule of tropomyosin for 7 actin subunits, or one molecule of tau for 4 tubulin subunits, or one molecule of cofilin for 1 actin subunit. So basically you need to stabilize, or whatever the desired effect is, you need to pretty much coat your filament or your polymer with these proteins. You have to coat the whole thing. But a capping proteins, you can just put one molecule of CAP-Z on the whole actin filament and it will stabilize it. Does that make sense? If you think about how these different side binding proteins work, usually it's binding to the side and it's doing something right there where it's bound. But because you have addition and subtraction at the ends, when you mess with the ends by capping them, you are going to stabilize them more readily. Make sense? Okay. So they're way more effective than side binding proteins. So an example to think about is in muscle cells, right? Muscle cells, do they have long-lived actin filaments? Or are they always disassembling? Sorry, I'm just laughing at myself. Are your muscles always wasting and then coming back and wasting? No. So they have fairly long-lived actin filaments, and so they're capped at their plus end with CAP-Z and at the minus end by tropomodulin. Right, so what does this do to actin filament dynamics when you've got them capped at both ends? Is it highly dynamic or not changing at all? Right, okay. Everyone can think about muscles, right? Are your muscles changing all the time? Well, I mean, if you're on steroids and you're at the gym all the time, I don't know. Anyways. Okay, so let's talk about proteins that bind to microtubulins. So we just talked about proteins that bind to actin, ends of actin. These are proteins that bind to microtubulins, and they can influence the frequency of catastrophes and rescues. And so you're going to get confused here because I'm going to say the word MAP. But remember, MAP just means microtubule associated proteins, and there are different ones that do different things, so just keep that in mind. A MAP protein called XMAP215, it binds and stabilizes, so it binds to the ends and it stabilizes microtubules, so it suppresses catastrophe. And the result is longer, less dynamic microtubules. And then there's this catastrophe factor, which you're not going to really forget what that one does because it's called catastrophe factor, right? That's destabilizing, and it's going to bind, and it actually kind of peels away the microtubules from the tubular chains from the microtubule, and that increases the frequency of catastrophes. So you end up with shorter, more dynamic microtubules. And when you think about how there's this competition between MAPs and catastrophe factors, and then there's just the regular dynamics of GTP hydrolysis resulting in catastrophe, you can think back to what the cell looked like when I showed you guys pictures of microtubules moving, bless you. And you saw they were constantly in and out, right? Constantly moving. So you have to think that, well, all of these proteins have a lot to do with it, right? Yes?
So for capping proteins, this is for microtubules or tubules. So what was for actin? Could you give an example of that?
CAPZ.
Capsule.
Yep. So the question was, this is for microtubules. What was the example for actin of capping proteins? That was CAPZ. Yeah. Okay. And you can actually also think of ARP as a capping protein, right? Because it kind of caps at the minus end and nucleates, and then you have growth. But it is, in a way, if it's still bound, capping. Okay. Questions?
The catastrophe factor is a protein that associates with a microtubule. Is it not a MAP?
It is. It would be under the category of MAPs. Yeah, I think MAP. So that's where I just warn you about using MAP to describe anything. If you ever use MAP to describe anything on an exam, follow it with a MAP protein that does this. So I know if you're talking about stabilizing or destabilizing, because MAPs frequently are used to describe all microtubule-associated proteins. So that's a good question. So I warn everyone. If on the exam you just write MAP, you'll get it wrong if you don't explain what you're talking about. Now that doesn't mean you need to say XMAP215 and MAP3C528 or anything like that. Just tell me what it's doing. Okay. Plus tips, plus end tracking proteins. So they bind to the plus ends of microtubules, and then they dissociate when microtubules shrink, which is very cool. I think one of those initial videos I showed you that was just showing microtubules growing, it actually was using an antibody against one of the plus tips. I forget which one. I think it was EB1, actually. And it only binds when it's growing outwards. So what you saw is just movement out. You didn't see the back. You only saw out because it dissociates when microtubules shrink. So catastrophe factors and XMAP215 are plus tips that modulate growth and shrinkage. So this is where things can get confusing also. They're all MAPs, and then they're also plus tips because they bind to the ends, right? Plus end tracking proteins bind to the ends, and they modulate growth and shrinkage. And then there are other plus tips such as EB1, which control microtubule positioning. And I don't know if we're going to go much into that. They're essential for mitotic spindle positioning in yeast. Okay. Maybe you're thinking, woo, that's a lot. I'm scared. No, don't be scared. Just do what I said, okay? Draw it out. Place all the parts where they go. Note on it. This does this. This does this. Beware, they're all MAPs. Whatever you need to do. But start to organize it. If words work for you, organize it in a hierarchy with different categories. So you know what's doing what with actin, what's doing what with tubulin. Draw it out, show things coming and going, whatever you need to do to put it somewhere so that it's not just a bunch of words. Okay.